Hello, I will talk about collagens today. We will go through the structure and function of collagens, and I will also introduce the synthesis. I put a Word file for you in the basic metabolic pathways folder, and I also found a couple of useful videos if you want to spend a couple of minutes to go through some animations about collagen, collagen function, collagen synthesis. I actually wanted to start with the definition of fibrillar proteins versus globular proteins, because collagens that I will talk about today are fibrillar proteins. So their structure and function is very different from those proteins that you learned about until now, including enzymes or hemoglobin, etc. So extracellular fibrillar protein, one of them is collagen, and I will talk about the structure, classes, function, synthesis, and a list of a couple of disorders that are related to collagens. Let me just say a couple of words about how important it is to understand the structure and function relationship in proteins. Proteins are playing either structural or dynamic functions in our body, and the native structure of the protein actually equals to their active conformation. So whatever is coming of the gene, the protein, nascent protein can be modified and it is folded into a structure that is designed to support the function of the protein. The structure is usually dynamic and it can be changed with interaction with other molecules. This is what you saw in the globular proteins for example, in hemoglobin, that if hemoglobin binds a molecule, that it can change the structure, and then it can thus bind oxygen or release oxygen. It's very, very pertinent, and you learned a lot of examples of enzymes when the different substrates can bind to the enzyme in a specific site. But if there are regulators, other ligands binding to the enzyme, their substrate binding can be halted, inhibited, or it can be promoted through conformational changes of the protein. So the change in structure of the protein influences its function. It's also true if the DNA has a mutation, which the DNA which is coding for the protein, it can lead to change in the protein itself, which can change to the structure of the final conformation, and thus it will change in function. Again, you learned a lot of examples about how genetic modification of proteins can alter the function and cause certain diseases. For this particular set of information, of course, I assume that you know from your pre-studies something about protein building amino acids and the character of their side chain, how they can interact with each other, and how the protein folds, what is the primary, secondary, tertiary, and quaternary structure of the protein by definition. So let's compare globular and fibrillar proteins. As I said, you learned a lot of examples of globular proteins that sometimes play roles in the structure of the human body, but most of the time they have dynamic roles, they have functional roles. And this particular one is an enzyme which binds a substrate, changes conformation, and catalyzes the turnover of the substrate or substrates to products. They have a spherical shape, this is why we call them globular proteins, and a variety of secondary structural elements, alpha helices, beta sheets, that are folded into a three-dimensional classical globular form. They are usually water soluble, which means that the outside of the protein is built of amino acids that have side chains that are polar or even charged, because they need to live in a very polar aqueous environment and fulfill their functions there. But compared to globular, the fibrillar proteins are usually rod-shaped. They have repeating secondary structural elements, which means that majorly it is the same repeatedly. Usually they are high molecular weight molecules, and they have low water solubility. And most of the time they play structural roles in building the cells, building the body. I will focus today on one of the extracellular fibrillar proteins, collagens. But I wanted to remind you that you actually learned about one of the other extracellular fibrillar protein, or extracellular protein, which we called elastin. You may recall that it's very important to be deposited in extracellular matrices to give elasticity to the tissue. So I wanted to remind you too, for example, the blood vessel walls, the uterus, and for everyday work, the lung contains elastin. But also the structures, all of our internal organs contains collagens. Collagen is actually the most abundant protein in our body. Up to 1/4 of the total proteins is a kind of collagen. I just brought it here for you as an example, and I will not hold responsible you to remember these numbers. But if we take the dry weight of different tissues, then you see that we can go from 4% in the liver till 90% of the demineralized bone composed of collagen. So it's very important in every organ, every tissue, extracellular matrix to contain a kind of collagen. But how much is there? It's variable. Now, you will learn a lot about the function of collagens in this block, because collagens build bone, cartilage, ligaments, intervertebral disc, etc. that this block will focus upon. However, you already learned about a kind of collagen in a vital gases block, or probably it was the vital fluids, I am sorry, when you learned about kidney basement membrane and Alport syndrome, and that was one of the collagen type 4, that we went into analyzing the genes, the structure of the protein, how it builds that particular meshwork, which is very important participant in the filtering capacity of the kidney. Organs does, because every organ and every tissue has a kind of collagen, it very much depend on normal collagen structure. So a couple of examples, which I will not go through in details in this lecture, but just think about it. If collagen is there in every tissue and organ, if the collagen which is synthesized is not normal, there will be clinical consequences, structural and clinical consequences. So for example, if there is a problem with collagen in cardiovascular organs, then aneurysm, heart valve problems, etc. can happen. In bone, which is, you remember, 90% of the dry weight is collagen, bone fragility is one of the early signs that there is something wrong with the collagen structure. If we look at the skin, poor wound healing, hyperextensibility of the skin is a sign of collagen disorders, while in joints, the hypermobility and arthritis, development of arthritis that you can potentially expect with abnormal collagen structure. Eyes, it's in certain collagen disorders, definitely the eye and the ear will be involved, so dislocation of the lens, or regarding the ear hearing problems will be noticed. So what is so interesting and important about the amino acid composition and later on about the structure of this collagen molecule? As I said, collagen is everywhere, but what is important to remember that everywhere, where whatever type of collagen we talk about, the amino acid composition is has certain conditions that the collagen for normal structural formation need to follow. For every collagen, it's important to have 1/3 of the amino acids being glycine. Proline is also very abundant in collagen, about 13% of the total amino acids are proline, and about 9% are modified prolines. So modified proline means hydroxylated in different position, or lysine is also hydroxylated. Those are, of course, post-translational modification because hydroxyproline, hydroxylysine are not part of the 20 basic amino acid that build the structure of every protein. But post-translational modification can modify amino acid side chains, residues, and it gives a huge variety of proteins that we can produce. Now, what do you need to remember? Please remember this, it's important. This hydroxylation, which is very important for collagens and also for other proteins, the post-translational modification is done by two hydroxylase enzymes. One is called proline hydroxylase, obviously places hydroxyl groups to prolines, and lysine hydroxylase. Both of them are working with vitamin C. Thus, if you don't eat enough vitamin C, we cannot synthesize, of course, that in our body, then there will be consequences that this hydroxylation will not be placed on proline and lysine, and there will be consequences in the three-dimensional structure of collagens. So please eat your vitamins, eat a vitamin C, otherwise scurvy can develop. So what is the primary structure of the collagen? The primary structure of the collagen is actually a helical form. One of these three helices, which build the tertiary structure of the collagen, is called a polyproline 2 helix. This is the primary structure of collagen. What is typical of the collagen that in every third residues a glycine should be placed. And then proline and hydroxyproline are frequent. Hydroxylysine also is important to be built into the collagen. Why are these amino acids so important? So glycine, if you remember the structure of glycine, it is the smallest amino acid. And there is no side chain. Majorly the side chain is a single proton. So why is it important? Because when three of these helices come together to build the superhelix, triple helix of collagen, they need to get very close to each other. If there would be a larger side chain than glycine, that they would keep the triple helix apart, the helix, the three helices apart from each other. Thus, a good and strong triple helix could not be formed. Why is proline important? Proline is a helix breaker, which means that regular alpha helix cannot be formed. It again is important because when regular alpha helix is formed, it is winding around itself, and it would not give an opportunity for three of the polyproline helices coming together for the superhelix structure. Hydroxyl groups placing them in proline and lysine is important because hydroxyl groups can bound to each other through hydrogen bonds and to other amino acids, and it stabilizes the triple helix. And hydroxylysine also can accept glycosyl groups, which will decorate the surface of the triple helix, and it will give the opportunity of the extracellular matrix to be special, to bind to additional molecules to make the matrix strong. So when these particular polyproline type 2 helices are built, the three of them need to get together. And this is an indication of what I told before about the importance of glycine. So this is an example that glycine should be in every third residue, every third amino acid in the polypeptide chain. And if it is not a glycine, for example, this is an alanine, with still just a tiny little short side chain, it will keep the distance from the other two polyproline helices, and thus the triple helix cannot be formed tightly as it is needed for us. So what is keeping together three of these polyproline helices to build the superhelix or triple helix tertiary structure of collagen? Majorly, these are hydrogen bonds that are formed among the three helices, and one of the player in hydrogen bond formation is hydroxyproline, which also plays role in forming hydrogen bonds. Of course, one single triple helix actually never exists alone. What is very interesting about these collagen monomers or collagen triple helices that they are formed intracellularly, but they then released by the cell to the immediate extracellular matrix, and then they are forming a structure that we consider the final structural arrangement of the collagen in the matrix. So this example that I brought here is typical of fibril forming collagens. So you see here the triple helix, you see here the monomers, the triple helix collagen monomers that are released by the cell. And typical of fibril forming collagens that they self-assemble. So one triple helix binds to another one and another one and another one. Please imagine this in a 3D dimension. It is not a sheet. It is actually a big bundle of collagen fibril which is formed. So we call the arrangement, which is again self-arrangement. We don't need enzymes for that one. We call it quarter staggered manner parallel array, which means that if we take a collagen molecule, for example, here a triple helix collagen molecule, then it will interact with many other collagen monomers around itself. Again, please imagine this is three-dimensional with one quarter shift between the collagens. Now, what will it results, and this is what you see under the microscope, that these areas where triple helices overlap, they will show up as a light staining in the microscope. And when there is a gap between collagen triple helices, even if the others are there, it will show up with like a dark staining. So this staggered arrangement results in the striated appearance of collagen fibrils. So what we need to, so what is forming this network of collagens is without an enzyme, it's self-assembled quarter staggered manner, but this is still a loose assembly. For us to have a strong fibril, from the fibril forming or fiber forming collagens, we need to actually cross-link this particular self-assembled collagen monomers. Self-assembly, hydrogen bonding, ionic interactions, none of them are covalent, but to make a very strong fiber, we need to have covalent cross-linking of the collagen monomers already assembled into this is fibrils itself. So we need an enzyme for the covalent cross-linking, which we call lysyl oxidase. This lysyl oxidase enzyme is actually producing an aldehyde group from the amino group of a lysine residue at strategic locations within the triple helix of a collagen monomer. This particular aldehyde group is very reactive. It can react either with an other aldehyde, which we call aldehyde condensation, or the aldehyde can actually react with an amino group, which is shift base formation, if you were a chemistry major, and covalent bond will be formed. So you need the enzyme, which works with molecular oxygen and a copper ion. Forms the aldehyde. Aldehyde reacts with an amino or another aldehyde group from another modified lysine residue and cross-links these triple helices. So what to remember of that? So covalent bond formation is important for the stability of the collagen fiber, which is built of several collagen triple helices, and it is catalyzed by an enzyme which we call lysine oxidase. In some books, it is called lysyl amino oxidase, exactly the same enzyme, and it works with copper. So eating our copper and releasing and delivering to the cells and taking up by the cells and binding to this enzyme is crucial. Let's go further. So we have now a wonderful collagen network in the extracellular matrix that fulfills the function of the collagen. So depending on what the function of the collagen is, as I mentioned before, we have more than 20 members of the collagen family. Actually, I think we are up to 26 or 27 different members. But we can put them into three categories: fibril forming, fibril associated, and network forming collagens. So fiber forming collagens, including type 1, 2, 3, 5, and 11, and I will focus on type 1 more in the rest of the lecture, they are doing what I talked about before. The collagen monomers triple helices are released by the cells. They assemble in a quarter-struggle manner and cross-linked for strong fibrils. Fibril associated collagens means that when these fibrils are formed, these types of collagen, collagen type 9, collagen type 12, can bind on the surface of these fibrils. They modify the surface and they are playing a very important role in binding these collagen fibrils to another molecule of the extracellular matrix. So they build further the matrix. Network-forming collagen types 4, 6, and 7, you actually learned about. You remember kidney basement membrane, type 4 collagen, which is not assembling into fibrils, but they are assembling to networks, meshworks. You remember chicken wire. So this is a different assembly, but these are also triple helical. They are also assembling with each other to fulfill the function of this type of collagens. Now, how do we label and how do we know which collagen we are talking about? So let me introduce you to the labeling of type 1 collagen. So the family, the type of the collagen is labeled with a Roman numeral. So this is type 1 collagen. You remember, builds the bone and other tissues. It's Roman numeral 1. It's not I, it's 1. Okay? Now the triple helix is built of so-called alpha chains or polyproline 2 helices. These are not alpha helices, these are alpha chains. They are coming from different genes that produce this particular alpha chains, and depending on the type of collagen, we have one of the same or two different kinds or even three different kinds of alpha chains building the triple helix. So in case of type 1 collagen, we have two alpha 1 chains and one alpha 2 chain of type 1 collagen, which comes together to the triple helix. Now, as I mentioned, these are individual chains of different gene products that build the triple helix. So let me just a little bit expand your knowledge with, here are the fibril forming and the network forming collagens. I left out the fibril associated from this figure. And then type 1 collagen is built of two of the alpha 1 1 and one of the alpha 2 1 chain to form the triple helix. And it is mainly found in bone and tendon and skin and scar tissue, and some build the uterine wall, and some is important in the heart and the intestine. Very widespread use. Now, opposite to type 1, you see what the difference is? Type 2 and 3 are built of the same exact alpha chain. Three of them come together to build type 2 and type 3. Type 2 is very special in localization. The vast majority of the type 2 collagen is building our cartilages. At the end of the bones, you remember we have this cartilaginous, very important tissue, which gives the opportunity for the joints to move. Type 3 is more in the skin, blood vessel, uterine wall, intestinal surface, and so on. This is what you learned about. You remember the basement membrane, type 4 collagen. The type 1, type 2 is usually present, alpha 1, alpha 2 is usually present in every basement membrane. That you remember the alpha 3, alpha 4, alpha 5 composition was typical of the kidney basement membrane, and the problem, the X-linked inherited problem with the alpha 5 chain actually gave rise into Alport syndrome. So just a little bit of remembrance what you learned into vital fluid block in regard of kidney problems. Okay, type 5, type 6, I will not hold you responsible right now where they are, but you see that a lot of organs and tissues contain at least one, but many times more than one kind of collagen for their function. So I said that I will talk a little bit more about type 1 collagen. So type 1 collagen, which is distributed in bone and skin and internal organ walls and etc., but especially bone. 90% of the dry weight of the demineralized bone is collagen. There are a couple of other proteins. There are, of course, the mineral, which is making our bones stronger, is also participating in bone formation, which you already learned in this block or will learn in this block. So type 1, what do we need? We need two alpha 1 type 1 chains and one alpha 2 type 1 to form the triple helix. So you see the alpha 1 1 gene and alpha 2 1 gene, which needs to go through transcription and translation, and two of the alpha 1 and one of the alpha 2 type 1 will build the triple helix. So what is happening right when these polypeptide chains are coming off the ribosome? Post-translational modification will happen. So hydroxylation, you recall, by prolyl and lysyl hydroxylases. You remember the vitamin? Yes, it is vitamin C. Glycosylation of especially the hydroxylated lysine residue is going on. And when these post-translational modification and all the chains are ready, they self-assemble into a triple helix. So this is still intracellular. Now you notice something if you look at the figure. At the N-terminal and C-terminal of the polypeptide assembly, already triple helix, you have these non-helical regions. We call them propeptides, N-terminal and C-terminal propeptides. You may recall that many of our proteins are synthesized and released in a pro form, and then they are activated later by enzymes that cleave off the pro from the active form. Correct? So active form is not forming until the propeptides are cleaved off. Just recently you learned about proinsulin, which needs to be activated to insulin. You also learned about the enzymes of the digestive system. Pancreas is secreting them in a pro form, and then in the digestive tract, in the small intestine, they needed to be activated by cleaving of the propeptides. So this is what's happening for collagen also. The so-called procollagen molecule is released by the cell, and outside of the cell there are two different so-called procollagen peptidases, one specific to the N-terminal and one is to the C-terminal, to cleave the non-triple helical, more of a globular, big propeptides of the collagen. The result is getting a tropocollagen molecule. So why is it not happening intracellularly? Did you wonder about that? So what would happen if these two peptidases would be present in the cell and would cut this particular propeptides off within the cell? You remember what is the next step? The tropocollagen is self-assembling. So if it would be within the cell, then the collagen triple helices would assemble and would definitely kill the cell. So this is why procollagen is released. The procollagen peptidases are outside in the extracellular matrix. They produce tropocollagen. Tropocollagen is assembled in the quarter-straggled manner, and we have the fibril. This is self-assembly. We don't need enzymes from that. Now, what is next? To make this self-assembled, already fibril, many monomers of collagen triple helix is assembled in that, more stable. So this is when we need this lysyl oxidase. Do you remember what we need for the lysyl oxidase to work? Yes, it is copper. So lysyl oxidase, copper, and cross-linking, covalent cross-linking among the triple helices. Now I put something here. This cross-linking mainly happening in the so-called telopeptides. So when the tropocollagen is formed, there are tiny little couple of amino acid long ends that are not triple helical, that are a little bit more open. And this is where the enzyme lysyl oxidase will form the covalent cross-linking interchain. And this fiber now is strong, cross-linked, can withhold load, pressure, whatsoever. They will not fall apart. Now, collagens are very long-lived. It was calculated out that the type 2 collagen in the cartilage has a half-life of 120 years. Can you imagine? So some of the collagen that you were born with, potentially you will die with. It sounds morbid, I am sorry, but this is it. Half-life means 120 years, very long-lived, which means that this particular collagen is there to support the tissues, support the extracellular matrix, support and give shape to certain organs. It is in our best interest that it is a stable network and it will not be degraded every day, and we don't need to replace that network all the time. However, of course, there will be damage made, and then we need to repair that network of collagens. So we need to have some some enzymes that can actually cleave the damaged collagen and it can be replaced. So these molecules, these enzymes are called matrix metalloproteinases. Of course, called metalloproteinases because they work with metal ions. And the most important is calcium or zinc that they work with. So these are called collagenases because they can attack the triple helical collagen. It's very rare that there is a need for that, but it does happen because we do reform our extracellular matrices. Now, what happens when these MMPs or collagenases cut the triple helix? Then there the triple helix starts to unwind. And when this unwinds, the triple helix, we get so-called gelatins and other enzymes, which we call gelatinases, also fall into the matrix metalloproteinase category. They can degrade the collagen down to amino acids. So a question here, do you know what you eat when you eat jello? Yes. This is the denatured collagen, partially treated with collagenase and gelatinase, heat is disassembled, and then you get this gelatinous network. Usually it is made from pig skin, so vegans cannot eat jello. All right, now it's obvious when we have such a huge need for collagens, such a huge need to build networks for us, to build fiber for us. We always need to have the presence of normal collagens. So abnormal collagens can cause big problems. So just to understand how complex what you have to do to form the normal collagens, let's go through this again. What do you need to form normal collagen network or fiber? Activation of the appropriate gene or genes if you need different alpha chains to build a triple helix. You need to have that gene dictating that glycines and prolines will be there in strategic locations. Also, hydroxylation, post-translational modification must happen. We need then the enzyme, which is also a gene product. We need the vitamin for that. We need an enzyme for glycosylation. We need enzymes to remove the propeptides from the end and the C terminal of the collagen monomers. We need the self-assembly of them, which again is dictated by the primary structure, and form cross-linking, which is the right amino acid at the right position, the enzyme being there, and the cofactor, those copper needs to be supplied. So you need a lot of things working perfectly to have the perfect collagenous structure formed in extracellular matrices. So thus, anything can go wrong and you have abnormal collagens or abnormal collagen network formed. So it could be the result of genetic defect of the collagen genes, abnormal post-translational modification of the collagen, genetic defect in the modifying enzymes, you remember? Deficiency of the cofactors, vitamins or copper. This is vitamin C, that is copper. So all of this can go wrong and you can end up with a collagen-related disorder. So let me show you a couple of these collagen-related disorders. So most well known for the most longest time is scurvy, which is nothing else but vitamin C deficiency. It is known from ancient times. Hippocrates already wrote about problems. So of course, he didn't know vitamin C or the importance of that one. The connection actually between scurvy and ascorbic acid was not done until 1932. But beforehand, it was already discovered that eating fresh vegetables, fresh fruits is very important to keep the health of people. So, the sailors that ended up in scurvy in the Middle Ages, uh, uh, or the new era, they already knew that they need to bring vegetables and fruits and citrus and whatever to the road, because those salted fish would not supply enough vitamin C for them. So scurvy is rare. Scurvy can be an inherited disorder, though if that particular enzyme for which we need the vitamin C is not there or not working or cannot bind vitamin C or cannot do the hydroxylation. Now, infants might be prone to vitamin C deficiency and scurvy, which actually can lead to more problems that it can lead to in adults. If they are not breastfed and they live on boiled or pasteurized formulas, then vitamin C is degraded through heat treating of the milk, and it can result in scurvy. Now, why is it bad? Because if vitamin C is not supplied, then it can be a problem with the normal growth process of the bone. Of course, the other collagens will also be affected. So increased capillary fragility, hemorrhage, poor wound healing at the top of retarded growth. This is why it is even more important for children, because when we are adults, at least our bones are formed, but we can also develop joint damage, we can develop orthopedic problems, but this is typical of looking at losing the teeth or having hemorrhages all around because the vasculature is not built strong enough. Another set of disorders, which I will supply you with an additional lecture material, is osteogenesis imperfecta, and it is typical of type 1 collagen disorder. So just a couple of words, it is also called brittle bone disease, because you remember type 1 collagen is the collagen in the bone. So most of them are genetic disorders, well, all of them, and it can be either the alpha-1,1 or the alpha-2,1, the gene which encodes for the alpha-1,1 or alpha-2,1 polyproline chains can be a problem in the different subtypes of osteogenesis imperfecta or OI. Actually, this already tells you osteogenesis imperfecta means that imperfect formation of the bone. So these will be bony changes from starting in the fetal life. Most of the OIs actually can be detected early in the fetus, characterized of multiple fractures, bone deformity, and because collagen type 1 is also present in the cochlea and the sclera, defective, progressive deafness, blue sclera, defective dentition also are signs of some of the subtypes of OI. Now, this is a syndrome which we call Ehlers-Danlos syndrome, which means that not only one collagen but other collagen types can be affected by a problem with an enzyme or cofactor, and they are put into one big group which we call Ehlers-Danlos syndrome. Typical of them, weakened connective tissue. Some of them will result in thin, fragile skin or impaired joints or hyperextensibility of the skin. It is really very funny looking, and other deformities. So let me introduce you to just a couple of Ehlers-Danlos syndrome biochemical reasons. So this is type 4, 6, 7, and 9. And I would like you to remember of what is going wrong in these particular types of EDS. So type 4 is a type 3 collagen problem. And obviously, because type 3 collagen is very important in the skin and in some internal organs, that will be the symptom that you will see. So it is autosomal dominant mutation, which is the deficiency of type 3 collagen network which is formed. Type 6 in an autosomal recessive deficiency of lysyl hydroxylase. You remember it's important to hydroxylase some strategic lysyl residues to bind to each other or to with hydrogen bonds or to being further modified, glycosylated. So it can be a variety of consequences because this lysyl hydroxylase work with different kinds of collagen. So usually scoliosis, so some bone deformities, hypermobility of joints, skin problems, ocular injuries, because lysyl hydroxylase is important for almost every type of collagen modification. Type 7 is the inability from the procollagen, the N-terminal propeptides. Joint problems and skin problems, usually, which are obvious. But because this propeptide is not removed from some of the collagens in the bone either, there will be some problems with the strength of the bone. And type 9, as a last example, is an X-linked recessive deficiency of lysyl oxidase. It also has some subtypes, and I will go there in the next slides, but let me just say it's very important in skin, in visceral organs, but also skeletal deformity will be one of the signs of lysyl oxidase deficiency. So why I'm saying that type 9 has subtypes, because it could be not just that the lysyl oxidase, the enzyme, is not working so that the aldehyde residue will not be produced so that it cannot cross-link, but that there is the very important copper without which lysyl oxidase cannot work. So the subtypes are the actual genetic problem with the enzyme, or copper deficiency by nutrition, or copper deficiency because there is no good way of transporting copper, either extracellularly or intracellularly, or it is transported intracellularly, but it is not distributed well within the cell or among the cells. So there are a couple of subtypes: Menkes, kinky hair syndrome. It is really the wiry looking of the hair, or cutis laxa in cows, which are typical subtypes of type 9 EDS. Now I just put it here for the sake of saying that when you have vitamin deficiency, that will affect enzymes, correct? If you have, in this case, copper deficiency, it affects how the enzyme can work, but it can be acquired too. So if you take vitamins, but vitamins are degraded or bound away or stored and not released, if these particular metal ions are taken up but bind away and not released, that will actually create a vitamin or mineral deficiency. So this is what I meant, that it can be acquired. For example, in this particular case, a copper chelating drug which was taken by a patient on purpose. So it was penicillamine. It the baby that was born with acquired Ehlers-Danlos because penicillamine in the body of the mother kept copper away from the circulation, and the copper didn't go into the developing fetus to aid with the cross-linking of the collagen fibers, collagen triple helices. All right, so we talked about a lot of things in this lecture. I hope you remember some. So the collagens are the most abundant proteins in the human body. Common structure is triple helix, built of 3 alpha chains that can be the same, like in type 2 collagen, or can be different. And one example was the type 1 alpha 1, 2 copies, and alpha 2, 1 copy for the triple helix. These alpha helices or alpha chains are coded by different genes and depends on the collagen type, which of these collagen genes will be activated and how the triple helix forms. We have fibril-forming, fibril-associated, and network-forming collagens. Specific amino acid sequence, typical triple helical structure is common for all. Now, how we synthesize them? An example I gave it to you for the type 1 collagen. Intracellular events is the appropriate gene activation, transcription, translation of the proper alpha chain, triple modification, post-translational modifications, assembly to the triple helix in the form of pro-collagen, then taking it out into the extracellular matrix when the propeptides are removed, and then the assembly of the collagen network or fiber will happen extracellularly, and covalent cross-linking make the structure of the fibril-forming collagen stable. And then related to this, lots of requirements of genes and assembly and post-translational modification, catalyzing enzymes, etc. I went and I introduced you to a couple of collagen disorders. Thank you for your attention, and please listen to the next lecture, which will be specifically about osteogenesis imperfecta.